Glomerulonephritis causes pathological glomerular injury with considerable cell death and extracellular DNA release. This protocol enables the visualization of extracellular DNA at the site of release during pathological injury. The advantage of this technique is that it measures extracellular DNA from cell death in the kidney, as opposed to serum or urine extracellular DNA measurements as a surrogate marker for pathological damage.
The significance of this method is that both the extracellular DNA staining and image analysis are easily transferable to other autoimmune diseases such as rheumatoid arthritis and lupus. This method requires a basic knowledge of kidney histology. To enable the accurate detection of glomerular extracellular DNA, the researcher must be able to correctly identify glomeruli.
For extracellular DNA staining, first, heat the samples of interest in a slide rack in 60 degrees Celsius for 60 minutes, before immersing the slides in 2 40-minute changes of solvent in a fume hood. After the second immersion, rehydrate the samples 2 times in 100% ethanol and 1 time in 70% ethanol for 5 minutes per treatment. After the last treatment, use tongs to place the slides into boiling antigen retrieval solution in a pressure cooker on high for 10 minutes. After unmasking the antigen epitopes, transfer the pressure cooker from heat into a sink and immediately run cold tap water over the lid. Allow the slides to equilibrate for 20 minutes in the antigen retrieval solution, before washing the samples 2 times in 0.01 molar PBS on an orbital shaker for 5 minutes.